Welcome to section 10.2, Biotechnology in Medicine and Agriculture. In this section, we'll connect the DNA tools you've already seen to real-world uses. We'll look first at medicine, how biotechnology helps with diagnosis, treatment, and production of medicines. Then we'll examine agriculture, how biotechnology changes crops and farming. Together, these examples show how DNA technology impacts everyday life. By the end of this section, you should be able to describe biotechnology uses in medicine, including diagnosis, vaccines and antibiotics, gene therapy, and transgenic animals. You should also be able to describe biotechnology uses in agriculture, including transgenic plants, agrobacterium-mediated transformation, BT crops, and the flavor saver tomato. Biotechnology in medicine includes many approaches: diagnosis, gene therapy, vaccines, antibiotics, and even transgenic organisms or engineered tissues. Think of this as the medical toolbox that comes from understanding and manipulating DNA. Genetic diagnosis means testing DNA to look for variants linked to disease risk or inherited disorders. A well-known example is the BRCA gene testing program, which can reveal elevated risk for certain cancers. The goal isn't to predict everything about the future. It's to provide information for prevention, early detection, or targeted care. As techniques improve, we can test from very small samples with high accuracy. Modern biotechnology helps us produce vaccines and antibiotics more efficiently. For vaccines, cloned genes let us make safe antigen components rather than relying on whole pathogens. For antibiotics, microbes, including engineered fungi and yeast, can be optimized to produce larger, purer batches of medicines than traditional methods. Biotechnology scales up production and improves consistency and safety. Viral vectors are engineered non-infectious viruses used as delivery vehicles. In vaccines, they can carry genetic instructions that teach the immune system what to attack. Some COVID-19 vaccines used this approach. The same delivery idea is being adapted for therapies. In principle, a viral vector could carry a healthy gene into cells to compensate for a faulty one. Delivery and safety are active areas of research, but the approach shows why viruses can be powerful tools when re-engineered. Gene therapy introduces healthy copies of genes into patients' cells to replace or supplement faulty ones. It is still experimental, but has shown promise in clinical trials for diseases like severe combined immunodeficiency, or SCID, spinal muscular atrophy, SMA, hemophilia, sickle cell disease, and cystic fibrosis. The aim here is to treat the root cause, not just the symptoms. With recombinant DNA, microbes can make human proteins. Two classic examples are the hormone insulin as well as human growth hormone, or HGH. Instead of extracting these from animals or even cadavers, we program bacteria or yeast with the human gene so they can produce a protein that matches ours. This improves supply, safety, and affordability and shows how DNA knowledge turns into real medicines. Transgenic animals carry engineered genes that make medically useful products. For example, goats have been engineered to secrete atrin, antithrombin, in their milk, used as an anticoagulant. Other projects put lysozyme into goat milk to combat diarrhea. or even spider silk proteins for medical and industrial uses. These examples share a theme: use the animal's biology as a production system for complex human products. In agriculture, biotechnology creates transgenic plants with new traits, like pest resistance, herbicide tolerance, drought, or virus resistance, and uses biological tools to transform plant cells. The key benefits are protecting yields, reducing chemical inputs, and improving the shelf life or even nutrition, depending on the trait. Transgenic plants are made by inserting one or more new genes into the plant's genome. Those genes direct the plant to make new proteins, perhaps an enzyme that detoxifies an herbicide or a protein that deters insects. The result is a plant with a novel trait that farmers can rely on season after season. One major method for plant transformation uses the bacterium Agrobacterium tumefaciens. In nature, this organism inserts its own DNA into plants. In the lab, we replace the bacterial genes with our gene of interest and then let Agrobacterium deliver it into plant cells. Those cells are then grown into whole plants that carry and express the new gene. Bt is a soil bacterium that produces proteins toxic to certain insects but that are safe for humans. When scientists transfer Bt genes into crops like corn or cotton, the plants produce their own insect resistant protein that can reduce pesticide spraying and protect yields. As with all resistance strategies, stewardship matters to slow down insect resistance in the field. The Flavor Saver tomato was the first genetically modified crop approved for sale in the United States back in 1994. It was engineered to slow softening and spoilage, which improved the shelf life and flavor of the tomato. Although it left the market a few years later, it set the stage for how genetically modified crops would be developed, tested, and regulated. And it sparked public conversations about safety, labeling, and consumer acceptance that continue today. Biotechnology has reshaped medicine and agriculture. In medicine, it enables genetic diagnosis, gene delivery, and the production of vaccines, antibiotics, and human proteins. And it opens the door to gene therapy and transgenic animals that manufacture complex drugs. In agriculture, it produces transgenic crops with traits like insect resistance or longer shelf life, using tools such as agrobacterium based transformation and Bt genes. The common thread is using our understanding of DNA to solve practical problems while also weighing ethics, safety, regulation, and long term impacts.